Hello guys, good afternoon. You are welcome to my YouTube channel, the SPC Tutorials, and my name is Dr. Joseph. Please, as you are watching this video, endeavor to subscribe, like, share, and comment. In today's video, we shall be treating AEB 412 called Comparative Vertebrate Embryology. Now, we've already trashed gametogenesis. Underneath, we have the oogenesis and the spermatogenesis. In today's video, we shall be looking at the second part of this course, which is what? Fertilization. This word is not new to us. Of course, we all know the meaning of what fertilization, but in this course we are going to be looking at the crucial parts of this topic, fertilization. Now, what is the meaning of fertilization? It means the fusion of the male and female gametes called sperm and egg respectively, to produce a diploid entity called what? Zygote. I said it is a fusion of the male and female gametes called sperm or spermatozoa and egg or ovum in the fallopian tube of the female organism to produce what? A diploid entity called zygote. Of course, zygote can undergo cleavage through mitosis to produce what? Daughter cells. Okay, but we are not going into cleavage this afternoon. We are treating fertilization. Now, since I've given us the meaning of fertilization, now fertilization has two independent processes. All right, one, we have activation of egg. Now, in this process, the egg gets ready to receive the sperm. That is, the egg will be activated. In that sense, the egg will get ready to receive the sperm. Number two is amphimexis. Please, another name for this amphimexis is sexual reproduction. It is a fusion of the nuclei of the male and female organism to produce zygote normally, and of course there is a transmission of genetic material from the parents to the progeny or to the offsprings. Now, since I've given us the two independent stages that occur during fertilization, now there are different steps that result to fertilization. The first step, I'll indicate it as A. The first step that leads to fertilization is the approach of the spermatozoon to the egg. Alright, the first one, which is denoted as A, is the approach of the spermatozoon to the egg. It means what kind of vigorous reaction does the sperm or does the spermatozoa or spermatozoon exhibit as it approaches the egg. Now, in doing this, we have to look at the various organisms that possess various kinds of fertilization. Now, take for instance the aquatic oviparous animals. All right. Of course, oviparous animals are animals that lay eggs, so they are called egg-laying animals. And fertilization is external, and they lay eggs. Now, and there are some of them are aquatic organisms, and some of them are terrestrial. But right now, I'm going to be restricting to just aquatic oviparous animals. Now, since they are found in the aquatic environment or the aquatic body, what happens? As soon as the male organism releases sperm, as soon as the male organism releases sperm, it is deposited inside the water medium, as well as the egg released by the female organism. Now, as soon as the both of them come together, there's this interaction. As soon as the egg is released, by the swimming action of the sperm or by the lashing movement of the tail, the sperm will swim towards the egg for fertilization, which means that as soon as the egg and sperm meets, they interact. As soon as they interact, fertilization begins to take place. Now that is what oviparous animals, egg-laying mammals. But as for the viviparous animals, now these are animals that give birth to their young ones alive. They do not produce egg. However, there are some that produce egg called monotremes, that is monotremes mammals. These are called egg-laying mammals. But right now, I'm not discussing egg-laying mammals. I'm restricted to just, let me say, humans. For instance, humans, dogs, elephant, whales. Okay. Now they give birth to their young ones alive, and fertilization is what? Internal. Now, in mammals, okay, where does fertilization occur in the female? It occurs in the fallopian tube. In the oviduct of what? The fallopian tube. Which means that as soon as the sperm is ejaculated into the body of the female organism, it moves by the lashing movement of its tail to the fallopian tube. Fallopian tube is the region of what? Fertilization of the egg. The reason behind fertilization is to form what? The zygotes. Okay, when zygotes is produced, it undergoes cleavage to produce what? Different daughter cells. Now, so this is just a brief introduction to what we have here. But you know, as soon as the egg is produced by the female, most animals, they do not possess attractants, which means that the eggs produced by female organisms, they do not have substances that attract the sperm, because the sperm itself has the ability to move using its flagella or what? Tail. But in some animals, in some few species, in some animals, such as there are two species of colonial coelenterids. So two species of colonial coelenterids. Now these species, when the females produce egg, they have what? They have attractants, which means substance that attracts the sperm towards the egg. Now, what are these examples? What are these animals whose females produce attractants? One, we have the Campanularia flexosa. That is XUOSA, please, because of space. Then number 2 is Campanularia cassiolifera, right? Campanularia flexosa and Campanularia cassiolifera. So these are the two species of colonial coelenterids. These organisms, they have their females possess what? Attractant. Now this attractant is called what? Gonangium or gonangium, as the case may be. Sorry. Now, for example, let us look at the Campanularia flexosa. Now, these organisms, they have a funnel tubule. These organisms, they have what? A funnel that a tube that looks like what? Funnel. And the females have this gonadium, which is actually the attractant. Now, how does it take place? Now, this gonadium of the female enhances the activity of the sperm. That is, it attracts the sperm. As soon as it attracts the sperm and the egg itself will not get deposited inside this funnel of these organisms. However, let me quickly say this. The activities that occur here are specific. They are species or specific, which means the species under Campanularia flexosa can only attract the sperm of these organisms, while these ones can only attract the sperms of these organisms, which means that the gonadium of these organisms cannot attract the sperms of these organisms. We say they are what? Species specific. They are species specific. Please take note of that. It is very, very important. Please, this gonadium is really an attractant. Yes, it actually attracts sperms. Apart from those two organisms, there are other mechanisms behind this gonadium. Now, when this gonadium is dissolved in seawater, in a pipette, all right, when this gonadium is dissolved in seawater in a pipette, what happens? It attracts the sperm. Yes, it attracts the sperm. Once you dissolve this gonadium in seawater in a pipette, what does it do? It will attract the sperm. That is for that. Another good example is the sea urchins. Sea urchins. These are echinoderms. Okay? When seawater is put in a petri dish, we are going to take some quantity of seawater in a petri dish and egg, that is seawater and egg. When they are put in a petri dish, after putting this egg, you remove this egg, then you insert the sperm. As soon as you introduce this sperm, what happens? The sperm will begin to locate that point you put this egg before. I recap: when seawater and egg are put in a petri dish and sperm is introduced, the egg, after removing the egg, what happens? This sperm that has been introduced will locate the point in which this egg was initially placed. Therefore, you say that seawater also plays a role in fertilization in certain animals like the sea urchins, which are the echinoderms. Okay, now, so that is for that. Is there any other points? Again, in fishes. Yes, who is that? Excuse me, please, excuse me. Hello, sorry for that. I have to attend to the person, please. Okay, we are back again. Now, I said that when I concluded by saying that when the egg of a sea urchins is put in, okay, when seawater and sea urchins egg are put in a petri dish and the egg is removed, you now introduce a given sperm. As soon as you remove that egg, as soon as that sperm is introduced, what happens? The sperm begins to locate the points where you place that egg before. Okay, now I said the eggs of fishes, they have what is called a micropyle. Eggs of fishes have what? Micropyle. It is this micropyle that the sperm penetrates through to meet the egg, which means without this micropyle, the sperm cannot locate the egg of these animals. Okay, what is that short description of the micropyle? The micropyle is a small funnel shaped opening through the chorion. Now, I told us in our previous class that egg envelope is called different names in different organisms. For example, now the egg envelope of fishes, fishes and tunicates, we call it what? Chorion. But the egg envelope of insects, mollusks, amphibians, reptiles, and birds, they are called vitelline membrane. But the egg envelope of mammals is called zona pellucida. They all have their names, okay? Now I said the micropyle is a small funnel-shaped opening through the chorion, which means it is through this micropyle that the sperm passes through to meet the egg for what? Fertilization. Which means that any egg, when any egg bearing the chorion is removed, the sperm cannot penetrate, meet the egg for fertilization. It means that in fishes, for fertilization to take place, the chorion, the eggs must have chorion, because the eggs must have micropyle on their chorion so that the sperm can successfully pass through it for fertilization. So what I've given so far is the approach of the spermatozoon to the egg, the approach. Now, under the approach of the spermatozoa to the egg, there are different things that occur. It reacts and it penetrates. Let us look at the reaction of spermatozoa. Definitely, as soon as the sperm gets to where the egg is, it begins to react. After undergoing certain reactions, it penetrates. Now, as I denoted the approach of spermatozoon to the egg as A, then under that A you have one called what? Reaction. It must be orderly arranged. It is very, very important. Now, look at this. When a ripe, or in the presence of a ripe egg, please, another name for ripe egg is what? Egg water. In the presence of a ripe egg, another name for ripe egg is what? Egg water. What is the meaning of egg water? Egg water means water having a ripe egg lying on it for some time, like in the same organisms, all right? It is water in which ripe egg of the same species are found lying for some time. Now, this egg, okay, gets stuck to by a sticky spermatozoa. Now, which means that this sticky spermatozoa will adhere itself to the surface of this egg. And the process whereby these spermatozoa adhere itself or themselves, since we're using spermatozoa, adhere themselves to the surface of these eggs is called agglutination. Agglutination. The process whereby these sticky spermatozoa adhere themselves to the surface of this egg is called what? Agglutination. Now, agglutination is made possible in the presence of a chemical called fertilization. I recap. Agglutination is made possible in the presence of a chemical called what? Fertilization. What is this fertilization? Now, this fertilization has its source to be this fertilization does not have its source from the egg itself. The source of this fertilization is not the egg itself, but the jelly surrounding the egg. The Lord is good. This course is going to be your slave very soon as time goes on. Let us follow it bit by bit. The grace of God is sufficient for us. Now, I said agglutination is that adhesion that this sticky spermatozoa exhibits with the surface of the egg. Then I said that is made possible by a chemical called what? fertilization. Fertilization has its source not from the egg itself, but the jelly surrounding the egg surface. Please take note of that. We are not done describing the fertilization. The fertilization is a chemical, sure. It is a chemical that is made up of glycoprotein and mucopolysaccharide. Right? I said the fertilization is a glycoprotein or mucopolysaccharide. Right? Fertilization is a glycoprotein or mucopolysaccharide. As a protein, it contains a number of amino acids. Of course, you know the monomers of protein are called amino acids, okay? So as protein, it contains a number of what? Amino acids, all right? But as mucopolysaccharide, it contains 2 or more monosaccharides. It contains 2 or more monosaccharides. Please, you have 2 C's there. What are examples of monosaccharides? They are glucose, fructose, galactose, glyceraldehyde, ribose, arabinose. Alright, all of these are what? Monosaccharides, and they are the constituents of these fertilization. I said that the fertilization is a chemical actually, and is a glycoprotein or mucopolysaccharide. As protein, it comprises amino acids. Then as mucopolysaccharide, it consists of monosaccharides, 2 or more monosaccharides, which are glucose, fructose, galactose, glyceraldehyde, ribose, arabinose. All of these are what? Monosaccharides. However, the type of amino acids and monosaccharides in the fertilization varies from species to what? Species, which means that the type of amino acids and monosaccharides present in the fertilization depends on the organisms involved. Please take note of that. Another substance, please take note of this. It is very, very important. On the surface of the spermatozoa, or rather the cytoplasm of the spermatozoa, all right, the cytoplasm of the spermatozoa has on its surface layer a substance called anti-fertilization. All right, I said the surface layer, okay, of the cytoplasm of the spermatozoa has a substance called what? Anti-fertilization. I said the cytoplasm of the spermatozoa has on its surface layer a substance called anti-fertilization, which means that the cytoplasm on its surface layer has the cytoplasm of the spermatozoa. The surface of it has a substance called what? Anti-fertilization. It's actually an acid. It is an acid protein. It is what? An acid protein having a molecular weight of 10,000 kilograms. So the weight of anti-fertilization is what? 10,000 kilograms. And the molecular weight of fertilizing is 300,000 kilogram. You can see now that the fertilizing is much more heavier than the anti-fertilizing. Now, the anti-fertilizing and the fertilizing, they undergo what? A certain type of reaction or interaction. Hope it's recording. Yeah, it's recording. Now, this is what is going to happen. As soon as the spermatozoon approaches the egg, what will happen? Okay, before I go into that, please, the anti-fertilizing and the fertilizing, okay, the anti-fertilizing and fertilizing, they exhibit a type of reaction, like the reaction between antibodies and antigens. So they call this reaction a type of what? Lock and key what? Reaction. Which means that the type of reaction that exists between anti-fertilizing and fertilizing is called what? A lock and key reaction. This reaction is similar to the one that occurs between the antibodies and what? Antigen. You know, clumping occurs between similar antibodies and what? Antigens. Please take note of that. Where is the border? Excuse me, please. Okay, we've established this fact already. I've given the molecular weights of the anti-fertilizing and the fertilizing. Please, as soon as the spermatozoon approaches the egg, there is this reaction. This reaction the reaction of the anti-fertilizing and fertilizing, right? The reaction of the anti-fertilizing and fertilizing accounts for the agglutination. The reaction of the anti-fertilizing and fertilizing accounts or is responsible for the agglutination of the spermatozoa to the surface of the egg. To the surface of the egg. So that is the first thing you should know. Now, what is the reason behind agglutination? Yes. Now, I said that when the sticky spermatozoa adhere themselves to the surface of the egg, you call that what? Agglutination. And it is made possible because of what? The fertilizing. Now, what is the reason behind agglutination? The reason behind agglutination is because the fertilizing has more than one active, okay, let us use the word reactive, more than one reactive spot. Take notes. All right, the reason behind this agglutination is because the fertilizing has more than one reactive spot. Now, this reactive spot will begin to adhere themselves to two or more eggs. As soon as they adhere themselves to the surface of these eggs, what happens? They will join them together, they will bind binding. All right, they'll bind this. Okay, it's sperms now. Sperm, not egg. Sorry, sperm. Sure. All right, they will bind these sperms together. Now, the binding of these sperms together result to what is called inactivation. The binding of these sperms result to what is called inactivation. Now, the reason behind fertilizing and anti-fertilizing is to prevent polyspermy, right? Which means that the interaction or the reaction that occurs between this fertilizing and anti-fertilizing is to prevent polyspermy. What is the meaning of polyspermy? Polyspermy is a process in which two or more sperms penetrate the vitreous membrane of the egg, which is never possible. It's not done like that. All of 100 million sperms produced per day. Produced per day. All right. Only a few will render the vitreous membrane vulnerable, but only one would fertilize the egg, which means that you see the reason here is very, very obvious. The reason for this reaction is what prevents two or more of sperm from entering the egg for fertilization. Now we've come to the end of reaction. However, the binding of some sperms together does not mean that all the sperms were successfully binded together. Some would escape. Now, the one that escaped will now undergo penetration, which is number 2. You see, it is an orderly manner. All right, it happens orderly, sequentially. All right, so it shouldn't be arranged zigzagly from the first to the second, never the second to the first. Now, the escaped one will now undergo penetration. The escaped one will undergo penetration. Of course, you and I know that for the sperm to penetrate the membrane of the egg of an animal in question, the sperm must exhibit certain characteristics. Take for example now the sperm of a mammal. Okay, you know the egg of the female organism is surrounded by what is called follicle cells. The egg of the female is surrounded by specialized cells called follicle cells. Now, for the sperm to be able to penetrate successfully, these follicle cells must be penetrated. They must be weakened by the acrosomal reaction that the sperm would undergo. Now, you know that the sperm has the head, neck, and tail. The sperm has the head, neck, and tail. On the inside of the head, it has what is called the acrosome. On the inside of the head, it has the acrosome. The acrosome has lysosome. And this lysosome contains a hydrolytic enzyme. This same head possesses the nucleus, having haploid chromosome. And of course, I told that this nucleus is rich in DNA, the oxyribonucleic acid. Now, because of this enzyme that the acrosome possesses, now I told us in our initial class that the acrosome is produced from the Golgi bodies. I did mention that. Now, the acrosomes are produced from the Golgi bodies. Don't forget that. So this is what is going to happen. As soon as the acrosome releases hydrolytic enzyme, as soon as the lysosome of the acrosome releases this hydrolytic enzyme, it renders these follicle cells weak. Or this acrosome releases an enzyme called a lysine. This lysine is an enzyme that dissolves the envelope that surrounds the egg. After dissolution, what happens? The sperm is able to pass through successfully. However, I told us that the first division of the tail is made up of what? The mitochondria, and the axial filaments. Axial filament. So, I said the mitochondria provides energy for the lashing movement of its tail, which means as soon as the sperm approaches the egg, the mitochondria must produce a certain amount of energy for the lashing movement of these sperm. Take note of that. That is, since we've been able to understand this part, let us look at other animals. Animals like mammals, oh god, animals like mammals, their eggs are not easily penetrated, which means that they have complicated what structures. Now, I told us in the initial class that the most complicated type of egg envelope is found in birds. Is found in birds. Of course, I aren't saying that birds are mammals. No, that's not what I'm saying. But I told us that all of the three types of egg envelope found in certain organisms, the one that is similar to that of the egg of birds is mammal. Now I gave us something in initial class. I did something like this. Now this inner layer is definitely the plasma membrane. Alright, this inner layer is called what? The plasma membrane. Why this other one that is close to this other one is called the zona pellucida. Now why these eggs found around it, they are called the corona radiata. Imagine that. For the sperm to penetrate the egg, you have here the ovum. For it to penetrate all these layers to meet this ovum, do you think it's going to be an easy process? Look at the layers, look at the barriers. How is it going to penetrate this? This is what is going to happen. Now, as soon as the sperm approaches the egg, another name for ovum is what? Egg. What is it going to do? It will render this zona pellucida weak. As soon as it renders it weak, of course the first layer is going to pass through is the corona radiata. As soon as it passes through this corona radiata, by the energy produced by the mitochondria, it gets into the zona pellucida. As soon as it gets to the zona pellucida, it dissolves it. Now, I told the dissolution of this zona pellucida is as a result of this lysine produced by the acrosome. Now, since the sperm has successfully penetrated the zona pellucida, what is the next one? It is what? The plasma membrane or the plasma lemma. As soon as you get the plasma lemma, then the action, okay, mission accomplished, because this plasma lemma is highly permeable. All right, it is not as strong as zona pellucida and corona radiata. As soon as it passes through this plasma lemma or the plasma membrane, it enters into the egg, and fertilization begin to what? Take place. Take note of that. Also in amphibians. Okay. Let me quickly say this. Nice one, nice one, nice one, nice one, nice one, nice one. Good. I made mention of lysine, that for this zonal pellucidar to be rendered weak, lysine must be what? Produced. However, the mammalian sperms, produce an enzyme called uh, mammalian sperms produce an enzyme called hyaluronidase. Now, this hyaluronidase will now break the hyaluronic acid. Mammalian sperms produce what? Hyaluronidase. Now, this hyaluronidase will break or render the follicle cells. Now, I told us that the follicle cells, they are found surrounding the egg, and these follicle cells, they are cemented together by this what? Hyaluronic acid. The first thing that it will do is to break this barrier that joins these follicle cells together. After breaking it, then it's able to penetrate the egg successfully. Don't forget this enzyme, hyaluronidase. What does it do? It breaks down this hyaluronic acid that cements the follicle cells together. Please take note of that. Then after which, the explanation I gave begin to take place. It penetrates the zonal radiata and dissolves the zonal pellucidar, and finally it gets to the plasma membrane, which is highly permeable. It passes through this plasma membrane and get to the ovum. Fertilization begin to what? Take place. That is for that. Then in amphibians, amphibians are frogs and toads. Yes, I know we know that already. In amphibians, which are the frogs and toads. Who is recording? Yeah, it's recording. In amphibians, which are the frogs and toads. Now listen and listen good. As soon as the egg is released, as soon as let me clearly say this. Let me know. Now the egg of amphibians is surrounded by a membrane called what? The vitelline membrane. Now, for the sperm to successfully penetrate or to get to the egg, it will definitely, or it has to pass through the vitelline membrane. Please take note, in the egg of amphibians, the sperm can never pass through this vitelline membrane, not until the egg gets to the oviducts. I will repeat, the egg of amphibians can never be penetrated if the egg has not gotten to the oviducts. To be specific, the lower region of the oviducts. Now, as soon as the egg gets to the lower region of the oviduct, the jelly coats is what? Laid. Therefore, as soon as this jelly coat is laid, alright, the sperm can now successfully pass through this vitelline membrane and begin to carry out the work of what? Fertilization. Please take note of that. It is very important. The egg of amphibians the vitelline membrane of the egg of amphibians can never be penetrated. not until the egg released gets to the oviduct, specifically the lower region of the oviduct. Then the jelly coat is what? Laid. Which means that we can also say that the jelly coat also enhances what? Fertilization. Fertilization. Because it is this jelly coat that has been laid that will permit the entering of what? The sperm, which means that this jelly coat also plays a role in the process of what? Fertilization. Take note, the jellied egg cannot undergo what? Fertilization. The jellied egg cannot undergo what? Fertilization, since it lacks what? Jelly. You see now it has been dejellied. All right, take note of that. It is very, very important. Is there any other one I'm yet to give? Now, since we've treated reaction and penetration, okay, before the sperm is able to pass through the egg successfully, now there are certain changes that occur in the acrosome of the spermatozoa. Now, that brings us to the next subtopic: changes that occur in the acrosome of the spermatozoa. Just follow it bit by bit. It is very simple by God's grace. So long you don't get distracted, just pay absolute what? Concentration. Now, changes that occur in the acrosome of the sperm. Changes that occur in the acrosome of the sperm. Does it mean that the acrosome will just release an enzyme like that? No. For this acrosome to release a particular enzyme, what will happen? The acrosome must undergo certain reaction. Yes, I said it. The acrosome must undergo certain reaction. Okay. Now, what is the first thing? One, the outer membrane. The outer membrane of the acrosome breaks. What will happen? It will break. The outer membrane of the acrosome breaks. As soon as it breaks down, the acrosome granules dissolve in fluid. All right. As soon as it dissolves in fluid, it releases lysine. You see that? For it to release this lysine, what must happen first? The outer membrane will break down first, and its granules are dissolved in the fluid, and therefore there is a release of what? Lysine. Number two, the inner membrane of the acrosome, the inner membrane of what? The acrosome will fuse with the plasma membrane. I don't call it plasma membrane, I can call it what? Cell membrane. I can also call it what? Plasma lemma. As soon as the both of them fuse together, they release what is called the inner membrane of the acrosome will fuse with the plasma membrane, then acrosome filaments, which are thin and long, are released. This is about 1.7 macro. 1 to 7, sorry, 1 to, is it 1 to, no. All right, so, yes, now look at this acrosome filaments released show that the sperm has been what? Activated. Now, an activated sperm has the ability to now pass through what? A given egg, however strong that egg may be. As soon as this acrosome filament is released, then it passes through it easily, but not all the egg of mammals do not have this acrosome filament. Take note of that. So, okay, the inner portion. Oh, all right. Okay, let me rub this one off. Now, I said the inner membrane of the acrosome will fuse with the plasma membrane. Oh. Okay, that is the second process, please. Now, this one is not found in the third process, in the third step. All right. The third one is the the posterior the posterior wall of the acrosome, the posterior wall of the acrosome will start to elongate. The posterior wall of the acrosome will start to what? Elongate. Then the elongation of this acrosome, the wall of this acrosome, will trigger the production of what? A thin, long filaments. And these are tubular. They are what? Tubular. These filaments are called what? Acrosomal filaments. Alright? Acrosomal what? Filaments. So these are the changes that the acrosome must undergo before a given egg is what? Fertilized. We are almost coming to the end of this class. Now, I told us something just now that as soon as this acrosomal filaments is produced, it can now successfully penetrate the egg. Now, in mammals, I told us that the acrosome, there's no acrosomal filaments in the sperm of a mammal. All right, which means that it penetrates. Okay, let me splash on my brain. Penetrate the plasma membrane and fertilizes. This marker is bad already, and fertilizes the egg. So these are the various changes that the acrosome must undergo before before the sperm is able to penetrate the female egg successfully. So at this point, we'll call it a day. Thanks for watching. Do have a wonderful day. Please endeavor to subscribe and share. Thanks for watching.